Hello and welcome back. In this lecture we will start Chapter 8, talking about DNA and its role in heredity. We will focus on DNA, why it is genetic material that determines the traits that we inherit. Sadly, we won't be watching the classic Jurassic Park, which also examines genes and the ability to clone dinosaurs. Interesting behind-the-scenes fact about this lecture, that Jurassic Park picture is actually from the book, not one I added for the sake of this talk. However, maybe I will watch Jurassic Park later. Anyways, moving on. By the early 20th century, a theory was developed that genes were on chromosomes. For this, scientists didn't know what the mechanism for inheritance was. Was it proteins? Mitochondria? Some other molecule? Evidence began to accumulate indicating that DNA was genetic material. Circumstantial evidence that DNA was genetic material was as follows. Scientists knew that DNA was present in the cell's nucleus and within the chromosomes. Also, they know that it doubled during the S phase of the cell cycle, and there is twice as much DNA in diploid cells than haploid cells. This led researchers to think DNA may be important. DNA was first isolated in 1868 from white blood cells. The researchers called it nucleum and proposed that it was genetic material. They really didn't have proof that it was. They knew it was composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. If you remember all the way back from our chemistry chapter, you know that these elements are found in all living things. Remember the sponge elements? Well, DNA contains most of them. Dyes developed in the early 20th century showed color when bound to DNA in dividing cells. Here you have a plant cell undergoing anaphase. You can actually see this with a light microscope, and if you haven't already, you will be seeing this in lab eventually. The dye allowed the amount of DNA to be analyzed. Cells in G1 contain half the DNA that cells in the S and G2 and M phase had. By examining the amount of dye these cells took up, scientists saw that all non-dividing somatic cells have the same amount of DNA, while gametes had half this amount. Chromosomes contain DNA. They also have proteins bound to them as well. So scientists had to rule out proteins as genetic material. In transformation experiments, it was shown that DNA from one strain of bacteria could genetically transform another strain. To look at if protein was genetic material, scientists used bacteriophages. Bacteriophages are viruses that infect bacteria. They contain DNA in a little protein. Experiments showed that when a virus infected bacteria, it injected only DNA. Since the viral DNA genetically transformed the bacteria, this showed more evidence for DNA being a genetic material. Here is a diagram of a bacteriophage infecting a bacteria cell. It injects its DNA, leaving the protein shell outside. The DNA genetically transforms the bacteria cell. Other experiments with mammalian cells showed that they could be transformed as well. By inserting a gene for antibiotic resistance, the cells are able to grow in media containing the antibiotic. Here's a brief diagram of that experiment. Cells in the left beaker had antibiotic genes inserted into them, while the beaker on the right had normal cells. When placed into media with the antibiotic, Cells with the resistant genes were able to live, while the ones without the resistant DNA died. With evidence mounting that DNA was a genetic material, researchers knew that DNA was a polymer of nucleotides. However, its 3D structure was unknown. By knowing the structure of DNA, scientists could understand how DNA replicated in direct protein synthesis. The structure of DNA was deciphered when many types of evidence were considered together. Rosalind Franklin performed X-ray crystallography on DNA and produced the crystallograph. You can see a picture of one of hers on the top right. Her picture of DNA suggested that DNA was helical and spiral in shape. Knowing the chemical composition of DNA also helped to determine its structure. Biochemists knew that nucleotides consisted of the sugar deoxyribose, phosphate group, and a nitrogen-containing base. As we discussed earlier in the semester, purines were adenine and guanine, and pyrimidines were cytosine and thymine. In 1950, a researcher by the name of Erwin Chargaff found that the amount of adenine always equaled the amount of thymine, and the amount of cytosine equaled the amount of guanine. In other words, the amount of purines equaled the amount of pyrimidines. Two researchers by the name of Francis Crick and James Watson combined all this knowledge of DNA to determine its structure. Franklin's X-ray crystallography convinced him that it was helical. Density measurements suggested that there were 2 polynucleotide chains in the molecule. Their model of DNA showed that in order for these molecules to fit together, it had to be antiparallel. Quick fun fact, Jurassic Park and Independence Day Hollywood star Jeff Goldblum played James Watson in the biopic movie of Crick and Watson's story. Pretty cool, huh? Here's Watson and Crick with their model of DNA shortly after they proposed the structure. Hopefully this is mostly review, but here is a picture of DNA, shown as a double helix with purine pyrimidines making up the interior of the molecule. Watson and Crick were the first to suggest that bases made up the interior, with the sugar phosphate making up the backbone of the molecule. Although we take this knowledge for granted, Watson and Crick, with some help, were the first to suggest it. Per Chargaff's research, a purine on one strand must be paired with a pyrimidine on another, making the base pairs the same width down the helix. Their model had 4 key features that are essential to the structure of DNA. The double-stranded helix had a uniform diameter, hydrogen bonds between base pairs held the chain together, and van der Waals forces between adjacent bases on the same strand also added to the stability of the structure. The 2 strands are antiparallel according to the model. As we talked about a few weeks ago, the phosphate groups are bound to the F' carbon of one sugar and 3' carbon of the next. Here's the structure of DNA. You have a 3' end on one side, as you can see in the top right here, and opposite that side you have a 5' end on the other strand. This makes the strands antiparallel. The outer edges of nitrogen bases are exposed in what are called major and minor grooves. These grooves exist because the helixes are not evenly spaced. On the right, you can see a molecular model of DNA. You can see the major and minor grooves on the molecule. These grooves are important for the attachment of enzymes that will help to replicate and transcribe DNA, as we will see later in the chapter. DNA structure is essential to its functions. It is the storage of genetic information, and its precise replication can occur because of complementary base pairing. Also, DNA is susceptible to mutations. Lastly, it expresses coded information in the phenotype. Alright, here's our last progress check to end this lecture. Please take your time to answer these two questions, and let me know if you have any questions on the material. Thank you for listening, and have a good rest of your day.